Thank you for having me. My name is Goldberg. I'm going to tell you about some of my postdoctoral work in which I'm using genomics to investigate plant-insect coevolution. So I think most of us are familiar with the concept of coevolution, but just to make it clear, it is reciprocal selection between two interacting species in which one is selecting on traits in the other and vice versa. This occurs in both mutualistic and antagonistic contexts. And a few known examples include a number of host parasite systems, both macro and microscopic, as well as a handful of obligate mutualisms, including ant acacia interactions and figs and the fig wasps that pollinate them. And so the systems that I work with are more difficult to look at because they are more generalized. And the reason that it's more difficult to assess the presence of coevolution is because there's a greater chance of interaction traits arising through either stochastic processes or selective pressures that are external to the interaction of interest. And when I say interaction trait, I mean as broadly as possible, any trait that is mediating the interaction between two different species. And an example of a not co-evolved pollination interaction is the corpse flower and the flies that pollinate it. At first glance, it may seem like it is potentially co-evolved, as the aroma of the flower is well suited to the sensory system of the fly and vice versa. But in fact, although the aroma of the corpse flower has likely evolved in response to selective pressures exerted by the fly's sensory system evolved to locate the authentic smell of rotting meat that guides it to oviposition sites. And so the plant that I work with is Datura rhytii, which is a solanaceous shrub native to the American Southwest and northern Mexico. It grows really well in disturbed soil, so it's common along roadsides and in human. It's also highly toxic, which gives it a relatively small insect community that is highly specialized for the toxins within this plant. And this has given the system a much higher likelihood of coevolution to be occurring. The main insect that interacts with Datura rhytii is Manduca sexta. It is both the primary pollinator of the flowers as an adult, and the larvae are one of the most devastating herbivores that this plant can experience. If left to its own devices, a single caterpillar could defoliate an entire plant. But there's actually a bit more to the Datura manduca system. Manduca is a highly diverse genus with, I believe, around 100 species. The genus is found all across the Americas, from southern Canada to Argentina and Chile in the south. And they use a diverse assortment of host plants. Most of them, but not all, are within the family Solanaceae. And there's actually two different species of Manduca that feed on Datura rhytii, Manduca sexta and its relative Manduca quinquemaculata. And so Manduca quinquemaculata has a much more limited geographic range. It's only found in North and Central America. It also has a much narrower range of host plants. And its sister species is actually the Hawaiian endemic hawk moth. Manduca sexta, on the other hand, has a Pan-American range. It has very closely followed solanaceous crop plants such as tomato, tobacco, and capsicum. And its sister species are a couple of Caribbean species. And overall, Manduca sexta tends to have a greater range of host plants. In fact, recently, within the past couple decades, it's been found to begin using a novel non-solanaceous host plant, which is quite unusual. And so this has led me to hypothesize that Manduca quinquemaculata is actually more likely to have co-evolved with Datura radii because of this more limited range and tighter association with a few species of host plants. And so I'm working on two different studies, the first of which is a comparative genomics study looking at Datura and Manduca coevolution. I'm working on de novo assemblies of the Datura radii and Manduca quinquemaculata genomes. Luckily, Manduca sexta has been a model species for decades and thus has already been sequenced a few times. And quite recently, within the past couple of years, a high quality chromosome scale assembly has been published. I'm also working on a de novo genome for another species of Manduca, Manduca rustica. This is a species that does not actually feed on Datura. Its primary host plant is, but it coexists with sexta and quinquemaculata in Arizona, where I'm doing the bulk of this work. And so because of this, Manduca rustica serves as an excellent negative control, as it has most definitely not co-evolved with Datura. And I'm also going to do a population genomics study of the Manduca Datura mutualism by resequencing both plants and moths from across Arizona and California. And so with the comparative genomics study, I'm looking for signs of coevolution using the reference genomes that I'm producing and are already available. A couple of the signatures of coevolution that I'm looking for are accelerated rates of evolution in the genes that underlie important interaction traits. And this will be relative to homologs in non-co-evolved species, as well as genes that are not involved in mediating interaction traits. Also, I expect to see some degree of synchronization in terms of gene duplication and loss events within the families of genes that underlie these coevolutionary interaction traits. Currently, I'm working on assembling and annotating all the genomes that I mentioned, and I'm using PacBio Hi-Fi as an assembler, and then a couple different annotation pipelines, depending on which is best suited. So currently, I've just about finished the Datura radii genome. The Hi-Fiasm assembly produced a total size of 2.2 gigabases, which is about what we expected. Most of the contigs are really, really big, or at least a handful of them are, the largest of which was over 200 megabases. And over 20 of those contigs were large enough to potentially be whole chromosomes. And I had a highly complete BUSCO score over 97%. The breaker annotation pipeline that I used was able to find over 56,000 genes, only using publicly available protein evidence. I do have some old mRNA data, courtesy of Daniel Hare, but I'm having some trouble using it for annotation due to its low quality and age, as it was produced about 11 years ago. And the repeat masker analysis found that over 85% of the genome is composed of repetitive elements, which is not unusual to see in a large plant genome like this. And so the next steps that I'm working on currently are to use the DuPipe and Café analyses to look at how gene duplication events and gene families have expanded and contracted within this genome. And so my second study, the population genomics study that I mentioned, I'm going to be testing some of Jeremy Yoder's models of mutualism dynamics to see how they're playing out in the Datura Manduca system. And so I'm going to, later this summer, collect both Datura and Manduca from 16 known Datura populations, ranging from Tucson to Sacramento. And these populations will represent a variety of climates, So the line there roughly approximates the road trip that I will be taking. And so these climates include both the Sonoran and Mojave deserts, coastal areas in California, urban Los Angeles, agricultural regions in the Central Valley, and more higher elevation populations in the Sierra Nevada foothills. And I anticipate to see 3 of Yoder's models present in this data set. They are not mutually exclusive. And so depending on the genes in question, you can see multiple of these models validated. So the first of which is mutual matching, in which there should be widespread stabilizing selection on very important genes in the system. And so an example of this could be the biosynthetic genes that produce the very important floral aromas that attract Manduca sexta. So in particular, there's a compound known as benzyl acetone that is innately attractive to Manduca moths. Naive ones, freshly closed from their pupa, will follow this compound to flowers. And of course, the olfactory receptor in the moth could also be considered a critical gene. Sanctions is another model in which I would expect to see localized selective sweeps on alleles that improve the effectiveness or avoidance of these sanctions. And so an example in this system would be certain plant toxins and the ability of the caterpillars to detoxify it. So ultimately, the purpose of sanctions is to reduce the negative effects of interacting with your mutualist. And finally, signaling and recognition, in which we would observe balancing selection on certain genes due to variability in the quality of the mutualist. So Manduca is not very consistent from year to year, and so their population density can vary. And so I wouldn't expect selection to be always constant throughout the entirety of the range every year. And with that, I'd like to acknowledge everyone who has helped me along the way, the Bronstein Lab, Matskin Lab, and Barker Lab at University of Arizona, the NSF for funding, and a few people who have helped me all along the way. And with that, I'm more than happy to answer questions.